welcome to Biological Life Sciences channel. Today in this video we are going to discuss Introduction to Biotechnology. As we all know, the term biotechnology was coined by Hungarian engineer Karl Ereky in 1919. It refers to the science and methods that permits products to be used from raw material and with the aid of living organisms. Biotechnology is a diverse field which involves either working with living cells or molecules derived from them, or applications oriented towards human welfare using varied types of tools and technologies. It means using of microorganisms, live organisms for the betterment of the society or humankind. It is also used in maintaining a good environment that our well-being. It uses scientific processes to get new organisms or new product from organisms. Large areas are there which includes many approaches and methods in science and technology. The biotechnology involves many disciplines or branches of learning, includes all areas of life sciences. Means the interdisciplinary nature of life sciences can be studied using this, as we know that biotechnology involves genetics, chemistry, biochemistry, food science, food technology, engineering, mechanical engineering in biotechnology, chemical engineering in biotechnology, biochemical engineering, electronics, and microbiology. Except that there are another subjects also, such as genetic engineering, molecular biology, forensic sciences, and all these subjects are included under biotechnology. So this shows us the detailed interdisciplinary nature of biotechnology. So what is the definitions? Let's see in short. In biotechnology, it is combined by two words, biology and technology. Biology is the study of living things, and technology is the use of science in solving the problem or making the products which are beneficial to mankind. So the combination of biology and technology, any technique that uses any organism or a part of organism to make the products or modified product. Biotechnology is the use of living organisms and their components in animal, food, and other industrial processes. As you can see in this example, the 14-month-old genetically engineered salmon, that is left side, and the standard salmon. So you can see the difference between the biotechnologically modified organism. This is the biotechnologically modified salmon fish, which increases in size though they are of same age. So this is the product of biotechnology. There are few more definitions of biotechnology. Let us see what are they. Any technique that uses living organisms or substances from those organisms to modify a product or to improve a plant or animal for specific purpose. Means that whatever the change we are making will be beneficial to humankind. Specific purpose, such as example, to produce medicine and vaccines, increase animal milk production rate, increase animal size, reduce cost of traditional animal breeding. You can see here these are the various ways through which we can change the living organisms. Let's see the history of biotechnology year wise. 500 BC, the Chinese used moldy curd as an antibiotic to treat boils. So what these people were doing, they were using the curd which were rich in antibiotics produced by different microorganisms. These are the antipeptides which kills the bacteria, produced by bacteria itself. So they used the moldy curds in which the molds were grown for the treatment. The Greek practiced crop rotation to increase soil fertility. In 1675, after that, Anton Leeuwenhoek discovered the microscope, and it opened a new era for the research. In 1797, Jennifer inoculated a child with viral vaccine to protect him from smallpox. Later, it was discovered that it was caused by varicella zoster. In 1833, the nucleus of the cell is discovered. In 1855, the E. coli bacterium is discovered. Nowadays, you know that E. coli bacterium is a very important bacterium in genetic engineering. So, majority of the genetic experiments is done on E. coli bacterium. In 1855, the same year, Pasteur worked with yeast, eventually proving that they are living organisms. Earlier, they were thought to be non-living. In 1863, Mendel discovered factor, which were later called as a gene, while working with garden pea. He laid the ground for work of genetics, and he discovered the 3 laws of genetics. In 1902, the word Immunology first used. The term genetics was used in 1906. Bacteria and viruses called phages were discovered in 1915. Nowadays, we know that these bacteria and phages are used as a vectors in genetic engineering. That is an important part of biotechnology. In 1919, the word biotechnology was coined first time by Karl Ereky. In 1927, Muller discovered the X-rays that causes mutation. In 1928, Fleming discovered penicillin. That was an antibiotic and it was a breakthrough. In 1938, the term molecular biology was first used. In 1941, the term genetic engineering was first used. In 1942, the electron microscope is used as a characteristics viruses that infects bacteria called bacteriophages. This was discovered using electron microscope. In 1941, DNA is shown to be the building block of the gene. In 1949, Pauling proves that the sickle cell anemia is a molecular disease caused by mutation of substitution of amino acids. In 1953, Watson and Crick understood and proposed the structure of DNA. In 1954, cell culturing techniques was used. 1955, an enzyme involved in production of nucleic acid is isolated, that is DNA polymerase. 1956, the fermentation process is perfected and it is used for producing variety of products. 1960, the messenger RNA is discovered, that is mRNA, which is an important part of gene expression. In 1961, the genetic code is understood. 1981, the first genetically engineered plant is reported. In 1981, first mice is to be successfully cloned. In 1982, humulin, that is human insulin drug, produced by genetically engineered bacteria, first biotech drug is approved by FDA. This drug, that is human insulin, was produced by a company called as Eli Lilly in America. In 1984, the DNA fingerprinting technique was developed by Sir Alec Jeffrey. In 1984, first genetically engineered vaccine is developed. 1986, the first biotech-derived interferon drug for the treatment of cancer are synthesized. In 1988, US Congress funds the Human Genome Project, that is an ambitious project in the history of humankind. 2000, a rough draft of human genome is completed. Human genome sequencing means to know the sequence of whole entire human genome. In 2000 only, pigs are the next animal to be cloned by researchers to produce organs for human transplant. 2001, the sequence of human genome is published in Science and Nature. These two are very renowned journals. In 2002, scientists complete the sequence of pathogen of rice, a fungus that ruins enough rice to feed 60 million people annually. So this whole genome of this pathogen was sequenced. Another breakthrough in 2003, that is the Dolly, the cloned ship from 1997, is emphasized. So this was about the brief history, and still there is a development of biotechnology till now. Let's recall there are three types of biotechnologies. First is ancient one. As we saw in case of Chinese people, they were using moldy curd for treatment of different types of infections. So earlier history is related to food and shelter, including domestication of animals. Traditional biotechnology, that is classical one, is built on ancient biotechnology where people fermented promoted foods were developed like wine, beer, etc. And medicines were also developed from that. In modern biotechnology, that is today's biotechnology, it involves manipulation of organisms and using them in different format or different desirable format, and this is done through genetic engineering. Let's see the different branches of biotechnology or the way the biotechnology approaches. In modern biotechnology, there are different areas and it is still growing. Major areas are animal biotech, medicinal biotech, environmental biotech, industrial process biotech, plant biotech, and microbial biotech. Let's see one by one. In animal biotech, these are used to improve animals, mainly the domestic animals, and it is used to produce products that promotes human health. For example, increase milk productivity. These cows are genetically engineered for increase the milk producing ability of these cows, and these are transferred with the gene of high milk yielding varieties. And low milk yielding varieties is also able to produce lots of milk, and it is also engineered to produce human hemoglobin inside the milk. So this is a breakthrough, isn't it? Second advantage or second development in biotechnology is animal cloning. Cloning is copying animals' gene into many copies, starting with the embryo twinning, splitting the embryo in half. Advantages: it helps in preservation of endangered animals, studying the effect of drugs, etc., on duplicates, and improve agricultural production also. Creating test tube babies is one of the important outcome of biotechnology. In this, in vitro fertilization is done of collected ova outside the reproductive tract. Usually the fusion is done in test tubes, and the semen sample is collected from male and of desired quality, and ovum are removed from female, and these are fused inside a petri plate or test tube, and again this is implanted into the uterus as the diagram tells you here, and after the implant the baby grows there in the uterus and it is delivered after 9 months. So this is another breakthrough of test tube babies. Biotechnology has various applications in medical biotech also, and it is applicable to medical processes. Some examples are designing of organisms to produce antibiotic. The best example was the first organism discovered by Sir Alexander Fleming, that is Penicillium notatum, which produced the antibiotic penicillin, which helped lots of soldiers in World War to be cured from bacterial infection. It is done through genomic manipulations, and lots of drugs are made using the medical biotechnology, where the vaccines are developed using genetic engineering. Biotechnology has very important role in environment biotech also, solving the various environmental problems using bioremediation technique, using biological process to solve environmental problem. Biodegradation, it's a natural process of microbes in breakdown of hydrocarbon material. It helps in removing oil spills under used wastewater treatments, heavy metal removal, chemical degradation. The best example was the production of superbug by Indian scientist Anand Mohan Chakravarti, who used penicillium organism for the removal of oil spills. Industrial process also involves biotechnology. It is also known as industrial biotechnology and is biotechnology applied to industrial processes. An example is designing of an organism to produce useful chemicals. Another example is the use of enzymes as industrial catalysts to either produce a valuable chemical or destroy hazardous or polluting chemical. Here, different enzymes, different products, different amino acids, different proteins can be produced using the industrial processes, that is fermentation. White biotechnology tends to consume less resources than traditional processes and used to produce industrial goods. Let's come to plant biotechnology. It is the biotechnology applied to agricultural processes, that is plant. An example would be the selection and domestication of plant via micropropagation and tissue culture. This is one of the important advancement in biotechnology, that is plant tissue culture, with the help of which a single plant can be grown into number of plants in a very short time. Another example is designing of transgenic plants to grow under specific environmental condition or in the presence or absence of a certain agricultural chemical. For example, we can convert a regular variety into a drought resistant variety by transferring the genes from drought resistant variety in the organism or in the plant. So such kind of experiments can be done with the help of genetic engineering. Our hope in this case is green biotechnology might produce more environmentally friendly solutions than traditionally industrial agriculture. An example of this is the engineering of plant to express a pesticide, thereby eliminating the need for external application of pesticides. The best and the most important breakthrough in this regards is the production of Bt cotton, that is having the Bt gene, that is taken from Bacillus thuringiensis, that is able to produce a toxin, that is delta endotoxin, which is helpful in maintaining the plant's health, and there is not required of anything that is to be sprayed on the plant, and the plant itself produces that endotoxin which helps to get rid of the pest. Let's see microbial biotech. This is the last field in the biotechnology. There are many more fields that are evolving, but microbial biotechnology is one of them. It is the aspect of biotechnology which involves the use of microorganisms or their products. Microbial biotechnology is sometimes also referred to as industrial microbiology, which is an old field that has been given new dimensions because of the discovery made in the field of genetic engineering and in vitro manipulation of DNA molecules to generate new combinations of gene or genetic sequences. This helps in redesigning the genome of bacteria and making the bacteria produce very important polypeptides, proteins, enzymes which are required for humankind, and to place the gene under the control of different regulatory system and to induce specific mutations in the molecule. Thereby we can configure these organisms to produce a eukaryotic genes or eukaryotic proteins in bacteria, like Eli Lilly did with the insulin, that is production of insulin from humans into the bacteria. Animal biotechnology, it is also acts as a source of medicinally valuable proteins. Antibiotics can be produced from transgenic animals, and transgenic animals can be produced with having desired character. Animals are important model in basic research where gene knockout experiments can be performed. Designing and testing of drugs and gene therapies can be done, and for this animal cloning can be utilized. If you like the video, please share, subscribe, comment and subscribe the channel. Thank you.